So welcome back to a tale of genetic code, and uh, let's just get started with today's topic, which is model organisms. So we have seen in our previous videos that the tree of life is actually quite complex and includes many different kinds of animals. And if you just imagine the kind of diversity that living organisms demonstrate, then you would realize the task that it is to actually study them. And as such, we might not be able to actually study each and every one of them. And so we need to decide on few simple organisms that we can study and then extrapolate the findings that we get in these organisms, the things that we learn from them to the rest of the living organisms. And the fact that all of the organisms share a common genetic material, that is the DNA, and that the cells more or less perform in a similar manner, following the central dogma where information is transferred from the DNA to the RNA to the protein, and then the proteins to the functions, with a lot of differences, of course. But because the basic scheme of life, of the transfer of information and interaction with the environment, we are able to use certain organisms that we are comfortable studying and then taking what we learn from them and applying to ourselves and any other organism that we might not find very easy to study. Another reason is ethics. So, of course, it is nobody would want to become a subject for a very uh, potentially dangerous study where we might probably want to study the effect of radiation exposure or certain harmful chemical exposure on the functions of life. And we actually do this quite routinely with cell cultures, etc. Or, for example, if we want to say study the effect of genetic mutation on the stability of chromosomes, then we would want to expose culture of cells to radiation and see whether chromosomal breakage occurs. But you would not want to expose an entire organism for that. And you might want to study a simpler organism or cultures of cells for this purpose. So ethics are also involved when experimentation is with living organisms, right? So that is why we have to select an appropriate organism, a model organism, which would give us the most information with the least amount of pain to anybody, right? And then life cycle. So we as humans live about, say, 80 to 90 years of age. And ideally, for organisms on whom we are conducting genetic studies, we would want to see the outcome also, right? Uh, if the lifespan of the organism that we are studying exceeds our own lifetime, then uh, just studying the effect of a few mutations in that organism might take several generations of scientists to be working on that. And that is why organisms that we study usually tend to have very small lifespan so that multiple generations can be studied uh, in that organism. And then, especially for the smaller ones, the difficulty in genetic manipulation is also important. So if because there's such a wide variety when it comes to smaller organisms, they can actually be quite picky and choosy in selecting them based on such criteria. But now that we know why we cannot study every organism, let us just see what model organisms actually are. So nothing but creatures that are easy to study and they can be of practical importance in whatever we are actually trying to achieve. So basically, the fact that we are using another organism to study something means that we are not able to study the actual organism that we are interested in, and that is why it has to be of practical importance that whatever we are able to learn from this, we can apply it to the organism that we are actually interested in as well. And beyond the four things that we discuss here is a very important thing that uh, comes into light when you actually think about model organisms from a genetic point of view, right? So we all know Gregor Mendel. He is the father of classical genetics, who lived in the 1800s, I guess. Yeah. And you know what? The traits that he chose to study, they were not just random traits of the plants that he came across on a morning walk, right? He chose traits that were very distinct, that existed only in two varieties. For example, the seeds could either be round or they could be wrinkled. The plants could either be tall or they could be dwarf. The seed color could either be yellow or it could be green. So he selected these traits very intelligently because the more confusion you bring to study, the less uh, is the inference that you can make out of it, right? So if you have very minimalistic things that are the outcomes, if your outcomes are predetermined, then uh, you will probably be able to identify the factors that are affecting those things better, right? So, in fact, the term Mendelian trait has come to mean this. They are clear-cut and discrete characters that can be unambiguously assigned to one category or another. So that is the power of selecting the right model organism because if Gregor Mendel had selected a different plant or had selected different traits of the same pea plant, he would probably not have come across the laws of genetics at all. So that is the importance of model organisms in the study of genetics. And now that we have seen this, we can probably move to the actual note on model organisms. So we had seen this part. Model organisms are convenient to study, of practical importance. Yes. And yeah, so what we can see from this list is that model organisms range in their position on the evolutionary tree, right from prokaryotes all the way to human cell lines and plants and many different animals as well. And we will see a few of these model organisms in today's video, and I'll put out another video probably sooner than my usual pace to cover the rest of them as well. So let's start with E. coli. So E. coli is the prototypical bacterium that we are all aware of. And the guy who had coined the term operon had famously said that what applies to E. coli applies to an elephant as well. So that is what a model organism means. It means that the uh, the the things that we are studying, the basic mechanisms of life that we are studying in these organisms are so universal that they are applicable to E. coli and the elephant all the same. And what we know about E. coli is that it's a rod-shaped bacterium. It's a bacterium that inhabits the gut of mammals, including humans. And most of the time, it is actually a commensal. It does not do much. It just stays there and and minds its own business. But sometimes it can get really nasty and cause lethal diarrhea, especially in children and old people. But from the point of a model organism, it is a good one because it divides very, very fast, right? So a single cell cycle can complete in 20 minutes. That is a doubling. It's not just a single cell cycle, it's a doubling. So if you have, say, if you start off with 20 cells, then you can imagine 40 cells, 80 cells, 160 cells. And by the end of the day, you might have many, many more cells than can uh, probably fit the planet. And of course, that does not happen because the nutrients that you add are the environment is basically the limiting factor. But you get the idea that they are quite rapidly growing. And in fact, they are some of the most rapidly growing organisms among the model organisms that we'll be seeing in the subsequent notes. Apart from being very quick to divide, they are small and they have a simple genome. They have a single chromosome. They might have a few plasmids here and there, but it's a small round chromosome that we can cut in places, attach our own DNA of interest, and we can see whether that is disrupting any gene, whether the DNA that we are inserting is expressing. And we can do this based on like antibiotic resistance genes and such. So we know the genome of E. coli pretty well. It is fully sequenced. We know what each gene does and what disrupting each gene might do. So we have a fairly good idea about its functioning, and so we can uh, manipulate it to study any DNA of our interest, for example. And it is very inexpensive to culture. It basically grows on our gut. It can probably grow on a petri dish, right? And I had so apart from the studying DNA of interest, which I was giving an example of some time back, what we can actually do is put a piece of DNA that we are aware of what its function is, and we want actually the protein, right? So we put the DNA in E. coli, it generates the protein, and we use that protein for our purposes. So insulin, for example, can be produced in this way, right? But there are some cons as well. So this is a prokaryote. So if you want to understand uh, functions of a cell that are exclusively seen in the eukaryotes, then obviously you'll not choose E. coli. And in fact, even if you're thinking about, like I said here, you can make recombinant proteins, but you'll not be able to make any kind of protein. And the proteins you might be able to make in E. coli would actually differ in biological function from those that you can make in, say, a mammalian tissue. And that is because uh, the uh, the central dogma of genetics, of molecular biology, is somewhat simplistic. It is not as simple as DNA forms mRNA forms protein and protein does all the function. There is a lot of post-translational modification that actually happens once the protein has been formed, and that is actually quite critical to the protein's function. So the cellular machinery that is available in eukaryotic cells, and that might differ, in fact, between different kinds of eukaryotes, is obviously very different from any kind of machinery that is present in prokaryote, right? It's a single, it's a single structure. It is not subdivided into more modules inside of it for specialized function. So even though you might be able to produce some proteins here, they might not fold the same way that they might fold in the eukaryotic cell because of the presence of, say, different kinds of chaperones that help that protein to fold in a specific way, or different kinds of glycosylation that is required for it to reach a certain compartment where they are further modified, etc. So there are differences in what a prokaryotic cell can do as compared to a eukaryotic cell. And then there are differences in the gene structure as well, right? So we know that prokaryotes have a gapless genome, whereas we have hardly any uh, uh, genes as compared to the intergenic and intronic regions that are present in our DNA. So most of the bacterial chromosome is actually coding. And it's not just that, but a single mRNA might contain genes, might contain code for multiple proteins. So it is different in this aspect from what is present in the mammalian cells or in the eukaryotic cells. And then the regulation and function are also different because basically they are we have several different functions because the cell is more complex. And then, like we had seen that here, we were using the E. coli to generate proteins of our interest. But gram-negative bacteria have an outer membrane that actually prevents protein secretion. It wants to keep the things that the cell makes inside the cell. So it might cause especially even for the protein that we are able to synthesize, it might cause a decrease in yield uh, per unit volume of that protein. So there are other kinds of bacteria that scientists might want to use if that is what they're interested in. And then there are certain interesting forms of E. coli. So one I had just alluded to, saying that it can cause really nasty diarrhea that might be life-threatening. So this is the one. So you can see it has extra toxins and it can cause bloody diarrhea. It might be fatal. And one interesting fact is that this company, the Hudson Foods plant, had to recall 25 million pounds of ground beef because it was considered to be contaminated with this strain of E. coli, right? And there's this E. coli K20, K12, which has a plasmid that allows it to, kind of mate, transfer genome horizontally between two adult cells of E. coli, right? So there are several things that these might also teach us, and probably will not go into the E. coli chromosome today. Moving on to the next organism. So we'll go to Saccharomyces cerevisiae, which is the brewer's yeast. So already you can see some color here, even though this is a false color of a scanning electron micrograph. Interesting thing about this is that in its haploid phase, we have only 16 chromosomes and a DNA that is uh, just three times as large as E. coli. So I think E. coli has like 16 kilobases of DNA. I might be wrong at that. I'll just put the exact number in the as a text somewhere here. But uh, just uh, it has just three times the size of the E. coli's DNA, but only 1.5 times the number of genes. So we can already see that from E. coli to the yeast, there has come a gap between the genes, right? So we are having non-coding genes in this the cells of yeast because now it is a eukaryotic cell, and that seems to be the case in all the eukaryotes. And what are some good points about this as a model organism? So, point number one, of course, it's a eukaryotic cell, so we can do some things that we by definition could not do in a prokaryotic cell, right? And despite this being a eukaryote, it is a single cell organism. Its genetic manipulation is almost as easy as that of the E. coli. And while its growth is not as rapid as E. coli, it is still just 90 minutes, right, compared to uh, some other organisms that we'll be seeing in subsequent notes. Again, its culture is quite inexpensive, and even though it is a single cell organism, it has a larger genome as compared to the bacteria, and you can insert larger sequences if that you're interested in to study their expression, or maybe you want a protein out of it. So you can do those things because it's a larger cell and can carry a larger genome, right? And while unicellularity can be viewed as a good point as well, because it makes it easy to do a lot of things, you cannot study what tissue level organization and inter organ communication kind of things with something that is inherently unicellular, right? So I would not call it a strict con because you would not select this for studying how a multicellular organism works, but especially with respect to the multicellularity, right? But even so, just for the sake of it, we can call it a con here probably. And what is actually important is that the chromosomes are slightly different, especially the centromeres of yeast. They are somewhat different. In fact, the kinds of histones that are present in the centromeres, the way the cell cycle regulation takes place, the way the chromosomes separate, etc., they are different, and we might cover them in some future video. But these are some reasons. These are some things you cannot basically study in a yeast cell. It's not necessarily a con. In fact, I think that is uh, the beauty of them, that they teach us the different ways you can achieve the same thing. That is uh, basically depending on what you're studying and how different organisms can help you answer your questions, right? So, yeah, so here, because you can isolate the haploid cells of the yeast and culture them, you can study the effects of a mutation on a single gene because there's no partner gene to compensate for its function. That being said, most of the genes on this single haploid genome also are copies. So like 40% of these genes are actually copies. So you would not call them alleles because they're not present on the same locus, but they're still compensating for each other's function. So yeah, so they are interesting. These are interesting. And now we are coming to some really, really cool things. So you might not like the idea of roundworms, and they do cause nasty diseases in us, but they are quite interesting. So they are first of all hermaphrodites. They make their own sperm. They make their own eggs. They have a short lifespan of just 3 days. Not lifespan, but they take 3 days to reach maturity. They actually live for like a few weeks. And of course, you can keep thousands of these on a culture plate. Uh, the interesting thing about this is that the number of cells in an adult. Where have I written that? Yeah, so the number of cells of an adult roundworm are actually fixed. So if you take any adult roundworm and count the number of its cells, it's always going to be 959. In fact, this is so constant that you can take an embryo and you can see how it divides, and you can tell that okay, this cell is going to end up in this region of the adult worm. And when you know things in so much detail, you can actually uh, tweak the process of development and see what effect that is going to make on the development of this multicellular organism. So these are the things that you cannot obviously do with something like an yeast or bacterium. So that is quite cool. And the roundworm is what the RNA interference was studied in. And this is again an interesting topic that we might come back to in a later video. That being said, the genome of the this organism is 97 mega base pairs long, and it's carried on 6 chromosomes. So that is a lot of DNA as compared to the lower organism. So we're talking in kilo base pairs when it was about E. coli, and it was just 3 times as much. So still less than say about 1 MB maybe in the east. But now we have 97 MB of DNA in this Rhabditis elegans. And more fascinating than that is that the number of genes it has is very close to humans. So this is this 1 millimeter long organism wriggling around in soil, eating bacteria, and it has the same number of genes that humans do. So this is a parallel to what we spoke about in plants, in the previous videos where they have so many more genes. They have like 40, 50,000 genes, and we have no idea what they are doing. And you see this worm, it has all the number of genes that we have. And despite it having these number of genes, actually, if you see the amount of DNA it has, that is out of proportion. That means the gaps between genes are quite larger. And you can see that there are 4 intervening sequences that we call introns in between the exons of these genes. So that is Cinorhabditis elegans for you. Now coming to the famous humble fruit fly that that has done that has given us quite a lot of insights in the workings of the human beings, maybe even human beings, because this is a model for development, for studying development as well, not just development, but also differentiation, signal transduction. Now that these organisms are so complex, they have behaviors, so you can study behavior here. Yeah, so this is the fruit fly. So it has again a short life cycle, 2 weeks. So now you can see that we had started off with 20 minutes at E. coli, right? And then we came to 90 minutes for yeast, a few weeks for the roundworm, although it matures in 3 weeks, 3 days. That means it can lay eggs in 3 days. So you'll get the newer generation much faster than its lifespan, which is quite larger. So one point I think I forgot to mention is Rhabditis elegans is that it can also be used for studying aging because the number of cells, what each cell does, where it ends up, and you know that they have a life cycle of say 3 weeks, and what are the factors that you can change to get it to live longer. And from 97, this is almost double the size of the genome here in Drosophila as compared to the roundworm. And all of this, it carries on 4 chromosomes. And yeah, this is again a funny thing. So you might think that that 1 millimeter long roundworm is a lower organism and that it probably is less complex as compared to this large Drosophila, which has all these structures. It has these large eyes, it can see things, it has wings, it can fly, it has so many limbs and a digestive system and all that, but it has fewer genes. It has only 14,000 genes. Uh, so, but then despite it having only 14,000 genes, a lot of these genes are actually uh, the kind of genes that humans do. So we can actually use these genes to study the effects of their loss in humans. So that is one advantage of Drosophila. So I think we can end this video here because it's already probably getting too long. We can cover the rest of the organisms in another video. Thank you.